Hello, histology fans. We get to do histology for the first time in, I think, 3 weeks. So, today's lab, we are going to look at models of muscles and histology of muscles. I'm actually starting with the model of the skeletal muscle, and we're not going to look at this model, but it's in your lab book. So, a skeletal muscle has these long parallel fibers. So here's one long parallel fiber, there's another one there. And it has this visible banding pattern. So I can see light, dark, light, dark. Oops, dark, light. So it has a visible banding pattern. We aren't going to look at the slide where we actually see the bulb. We're going to look at a different model that has this though. And you have a motor neuron that's connected to several fibers. So we have long multinucleated cells that have banding. So we have these peripheral nuclei. So you guys are going to be sent to a website and look at this for me. Now, cardiac muscle usually only has a single nucleus. So here's the single nucleus. According to your textbook, it could have two. I think I've seen some slides actually that have two. But generally, it has a single nucleus. And again, it is banding. So we have these light lines made of actin, followed by dark lines made of myosin. Then, you guessed it, light, dark, light, and so forth. Now, another thing you should notice is there are these discs called intercalated discs. Yes, drawing black on black, I'm sure works very nicely. And we have branch cells. So these intercalated discs mark the end of cells. They're a combination gap junction and desmosome. So calcium ions are actually going to be able to flow through the gap junctions to the neighboring cells. Calcium will flow through to the neighboring cells. Smooth muscle gets a bad rap. It's actually very interesting. It has these short, tapered cells with a single nucleus. So short, tapered cells. And remember I pointed out how you actually had one bulb and one end. Here along the axon, instead of one bulb, you have these things called varicosities. And the neurotransmitters are going to actually leave and wash over it, sort of like a local messenger. So, here's skeletal muscle. I believe I gave you the choice to either draw it or to take a screenshot and label it. It's whichever makes you feel better. I'm in a drawing mood, so I am going to draw it. Now, it has long parallel fibers. So here are my long parallel fibers of skeletal muscle. And it has a light-dark banding pattern. So I will make my striations like that. 30 minutes later, I'm done with my drawing, my striations, and I am going to put in some peripheral nuclei. And I'm going to put them black. Just because they are black, I'm so tempted to do them blue. Just throw a little color in, but it's not what the slide looks like. This is not art class, even though I claim it is. So, these are our nuclei. So, on the lab exam, what you look for, parallel cells. So, parallel cells. We can see the banding pattern here. I'll show you a better slide of this in a couple minutes. So, here's cardiac muscle. You can see the banding pattern a lot better in this. And cardiac muscle has these short, branched cells. And there are these intercalated discs that are separating them. So here are my branch cells. And I'm going to put the striations in. And I'm going to put the intercalated discs in. So here are the striations. I drew them differently. I suggest for the skeletal muscle you draw them this way as well. It takes a lot less time. And just try to differentiate the intercalated disc is thicker. So like here's an intercalated disc, and this is what you should always look for when you're doing the histology. Does it have it's striated, it's either skeletal or cardiac. Does it have these intercalate discs? Then it's cardiac. So here are branching cells, and then centrally located nucleuses, generally only one, so in each cell. So we'll put the nucleuses in. I put 2 in that one. Oops. So textbook does say it could have 2. Now we're going to draw smooth muscle. So depending on the slide or where we have it, you could have a longitudinal section of it where we have the cells facing that way, or a cross section of it where we're looking at straight on, sort of like a pen. Now when we get to the intestinal tract and the urinary tract and the reproductive tract and all those tracts inside the body, we'll actually see both a longitudinal layer and a circular layer where we'll get cross sections. So we're going to have fistiform cells. Now in the face-to-face class, I tell my students, make your drawings big so you are not drawing that much. Nobody ever believes me. So I'm making these nice and large. So there are these tapered cells with a central nucleus. So there, the central nucleus. And just because that took me 30 seconds, I'm actually going to use the highlighter to color it. And there is smooth muscle. That is all. Now we're going to look at the sarcomere under histology, and we are going to look at the connective tissue elements. Do you recognize this bone? It is the femur. And what we have is a tendon. Tendons are going to attach muscle to bone. Now, the tendon's dense regular connective tissue, it goes off into this thing called the epimysium. The epi means around. And the epimysium is dense irregular connective tissue. This week, trending on Reddit and Imgur, is the picture of this person with pulled pork, and it looks like the pork is just a solid piece, and they literally just touch it and it falls apart. That's what barbecue does, is barbecue is just breaking this connective tissue element, and then you're just left with the protein fibers, and that's why the pulled pork falls apart, if it is cooked correctly. Now, the inside of the muscle, you have the fibers arranged in these things called a fascicle. Now, the thing is, you need blood vessels going between them. So we're going to have blood vessels in here. And the connective tissue holding the blood vessels and holding the fascicles is called perimysium. So this in here would be perimysium, and that's just holding it all in place. Then around an individual fiber, you have endomysium. So how does it look on histology? Well, here is the slide down here. This here is the epimysium wrapping the whole thing. We then have perimysium wrapping the fascicles together. So this would be all perimysium. Then I could sort of see the endomysium on these. I swear I picked a different color. We could sort of see the endomysium, but on this power, I could definitely see, here's the perimysium, here is the single fiber, so this would be endomysium. I always make students do it on both the scanning power, so we can see the epimysium, or mysium, and the perimysium, and under high power. So to start our picture under high power, I'm going to actually draw the fascicles. And here's a fascicle, here's a fascicle. What do I keep saying about drawing size? Should I make it small or large? Yep, large. So there's the fascicles. Now the epimysium is the space out here. So I'm just going to do epimysium around one side and label it. Now to differentiate, I change the color. So the perimysium is what is in between the fascicles. Can I talk to you and draw between the lines? I don't think so. Oh my goodness, am I going to? So this would be the perimysium. Now let's draw the fibers in here. So these would be where the muscle fibers are. We'll label them fibers. And just for the sake of propriety, I'm going to actually draw in some of the fibers there. So on the lab exam, I'll give you a picture like this, and you have to identify the different connective tissue elements. So here's low power. So under low power, I am going to have muscle fibers, like so. And these are now individual muscle fibers. So, yeah, my drawings look the same, don't they? So the difference is that right there is this. So this right here is that. So you understand that these little circles in here are now these circles. So the perimycium I did blue, so let's do it blue around the whole fascicle. And then I believe I used a light pink for the endomycium up there, so I think that's the same color. Here's the endomycium. There it is. I don't know why I was afraid to write down there further, but so try to distinguish between the two. Maybe do these as actually screen capture so you could distinguish. Here's the perimycium here. Here's the perimycium in this picture above. Because, yeah, my pictures might look a little bit similar. So we are lastly going to look at the sarcomere. And we'll look at both the histology of it and we will look at the model. So in lab or in lecture, I believe I drew the sarcomere 20 times. We have a central M line with myosin filaments coming off like so. And then we have thin actin filaments between the myosin. And the actin is attached to the Z disc. So this is what it actually looks like on the histology slide. So I'm going to start by drawing or showing you the Z disc. This is the Z disc right here. So this line right there is the Z disc. So for drawing the Z disc, you just have this thin line. Oops, that was the highlighter in black. So you just have a thin line. Then pick a light highlighter color, such as, I don't know, tan. Make a small highlighter. So then we have our A band. our I band. So there is our I band. That is where you have only actin. So only the actin, that's where the I band is. Now the A band that some person named wrongly, can you tell I'm still irritated with that, would be between. So this right here is the A band. And then we have another Z disc. And we have, oops, that was not the same color. The I band right there. So let's make these I bands the same color. So we have I band, that's the light, the A band, that is the dark. And sometimes you can actually see the M line. So right here, can you see the green? Let's make it orange. Right there's the M line. I only have, or the H cell. I only have actin there. So that is the histology for the sarcomere. So that's our histology. I don't have time to do another one even if I wanted to. So at least 2 questions identifying structures, at least 2 questions identifying what type of muscle it is. Thank you.